I'm Andrew Ramos, and welcome to my lecture on the function and history of environmental DNA. My article of focus was led by geneticist and zoologist Yuchang Wang out of the University of Cambridge. In this paper, the researchers reconstructed the paleoecology of the Siberian steppe tundra using environmental DNA, and it is environmental DNA that this lecture will be focusing on. Before jumping into the history of environmental DNA, I want to briefly describe eDNA sequencing and why it's possible. All organisms shed DNA into the environment. This can be by lost fur, feces, urine, skin cells, and even tears. As this DNA-bearing material becomes lodged in the soil accumulates over time. This allows scientists to drill sediment cores out of the earth and locate the DNA of organisms that lived at different times. Environmental DNA is a relatively young field of study. Its practice and use evolved as the technology behind it improved. In 1987, microbiologist Andrew Ogrum and colleagues introduced a new method to isolate extracellular DNA in sediment. Their target was the DNA from bacteria living in the soil. First, an alkaline buffer is used to extract cellular material from the soil. Then a bead beater is used to chop cells up. This separates them into different components such as DNA, RNA, and proteins. It is then processed further and sequenced. By the year 2000, eDNA was still limited to the realm of microbiology. Michelle Rondon and her team were seeking a technique that would allow them to pull even more DNA from the soil by limiting destruction of DNA while it is being filtered. Using a bacterial artificial chromosome, they were able to increase the amount and range of DNA recovered from soil samples. By 2006, eDNA made the jump from microbiology to marine ecology, from microbial life to the animal kingdom. Marine biologist Punalowski, Baduri, and associates decided to apply these same techniques to marine sediment in order to identify the presence of different nematode species. If we jump ahead to the year 2012, the term DNA barcoding was commonplace. Rather than targeting bacteria or a single plant and animal species, scientists were now sequencing all DNA fragments in a sample and using them as barcodes or fingerprints to identify as many organisms as possible. This process would continue, and by 2015, attention moved beyond simply identifying species in a sample to mapping the relationships of the animals and ecology of the environment. This was useful in fields such as conservation and wildlife management in order to monitor the wildlife in a region or the arrival of invasive species. Fast forward to the year 2021, and the field of eDNA had entered the world of paleogenomics. Vernat et al. Developed a technique to probe soil samples for ancient nuclear DNA. This allowed researchers to identify the presence of Neanderthals and Denisovans over a 100,000-year time span. These techniques limited analysis to humans or their close relatives. The progression of the environmental DNA field can be tracked through its historical interests. Its study of bacteria, to nematodes, to multiple species, to the ecology of these species, and finally to ancient hominins, led up to the current state of eDNA and the original study of focus in this lecture. In this study, Wang et al. Reconstruct the Siberian steppe tundra using environmental DNA. They were able to detect the presence of and relationships between ancient horses, rabbits, mammoths, humans, caribou, wolves, bison, and woolly rhinos. In fact, they were even able to document the extinction of the mammoth. As the mammoth's preferred habitat of open grassland and perennial fields were overtaken by woodland, they clung to the last patches of open habitat. eDNA shows that their last stand was likely north-central Siberia, where the last survivors died about 4,000 years ago. From studying microbes and dirt to reconstructing ecosystems tens of thousands of years in the past, the field of environmental DNA has come a long way in a short amount of time.